We will be going through several key systems in this course, from lung to kidney, neuromuscular, heart. All of those systems have the autonomic system influencing them. So I think it would do us well to consider broadly some principles on how the autonomic system works. So my task on this block is to do a kind of a whirlwind tour of the autonomic system. We know the autonomic system is made of the parasympathetic and sympathetic systems. So we will go through a broad sweep, and then we will loop back around as we work through each organ system and revisit the autonomic function, and in some cases go into a little more detail. So let's outline some principles regarding the autonomic system. This ANS, which is typically called, is controlling non-voluntary motor function in your body. So you don't have control over the parasympathetic or sympathetic systems in terms of dint of will. It's done below the level of consciousness. We call the part of the motor system that we do have voluntary control over the somatic nervous system. And that, of course, stands in contrast to the autonomic nervous system. And the somatic nervous system, again, deals with neurons coming from your motor cortices, and then they will go down, and then they will communicate with another neuron we call a motor neuron, and that neuron will leave the central nervous system, either the brainstem or the spinal cord, and will make its way to skeletal muscles. When it comes to the autonomics, the scheme is a little different. The autonomics, we have a communication with what we call a ganglion. A ganglion is a collection of neuronal cell bodies outside the central nervous system. If you have a collection of neuronal cell bodies within the central nervous system, and I'm defining the central nervous system as either brain, brainstem, or spinal cord, a collection of neuronal cell bodies within the central nervous system that has a similar function, we call that a nucleus. Now, nucleus is not in the sense of DNA, nucleus of a cell, but nucleus as in a collection of neuronal cell bodies that have like function. So again, collection of neuronal cell bodies within the central nervous system, call it a nucleus. If it's a collection of neuronal cell bodies outside the central nervous system, we call it a ganglion. That's singular. A ganglia is plural. So this is one of the differences between the autonomics and the somatic. Again, somatic, central nervous system, and this is either going to be the brain stem or the spinal cord. And of course, you have higher motor neurons. And let's just draw a little brain up here. There's my brain right there. Okay, so I'm going to have some motor neurons that will come down and will talk to other neurons either in the brain stem or the spinal cord, and then the brainstem or spinal cord. That's what this neuron here is symbolizing, and that neuron will send an axon out. It'll be a motor nerve, and it will then synapse onto a skeletal muscle. In the autonomic system, the scheme is a little different in the sense that we have, again, some neurons in the central nervous system, and we will learn in a bit where those actually are. They will then send an axon out leaving the central nervous system and progressing towards a ganglia. And this ganglia here is going to be synapsed upon, so it will release neurotransmitters onto this collection of neuronal cell bodies. And then those neuronal cell bodies will then send an axon out, forming a nerve, and they will then connect with or synapse on whatever their downstream target is. It could be smooth muscle, it could be cardiac muscle, it could be glands, etc. So again, it just depends upon what part of the autonomic system we're talking about. So this is a very good general contrasting scheme between somatic and autonomic. With the autonomics, it could either cause a contraction or it could cause a relaxation of smooth muscles. Again, it depends upon the system we're talking about, either the sympathetic or parasympathetic, or also could be talking about what organ systems are involved as well. And so we're going to find that it behaves differently. It depends upon again what part of the autonomics, and it depends upon the end organ system we're dealing with. So it could either cause again contraction of smooth muscles or a relaxation of smooth muscles, and we'll work on this as we go through. Another vocabulary that you're going to want to be aware of is that we have the term preganglionic fiber and postganglionic fiber. I believe you can figure out the pre and the post, and the pre and the post is referring to the ganglia here. So if we have two types of autonomic systems, parasympathetic and sympathetic, so we could have a parasympathetic preganglionic fiber, or we could have a parasympathetic postganglionic fiber, or we could have a sympathetic preganglionic fiber, or a sympathetic postganglionic fiber. So again, we have the para or the sympathetic para or the sympathetic that could be applied to preganglionic, postganglionic. A postganglionic fiber, by definition, is the axon. And let me say this as well, just to make sure we're clear. When I say fiber, in this context, I am speaking of axons from neurons. So we're not talking about the cereal All brand in terms of fibers. Okay, so we speak of fibers in terms of axons when we're dealing in this context here. So just make sure, I want to be clear with you on that. So if we're talking about a postganglionic fiber, then we're talking about the axon that is going to be leading to the end organ. If we're talking about a preganglionic fiber, then we're talking about an axon that is making its way to the neuronal cell body on which it will synapse. If you are confused regarding whether a fiber is preganglionic or postganglionic, all you have to do is ask the following question. And if you ask the question and answer the question correctly, you'll never get this mixed up. If I were to ask you, pointing to a fiber, is this preganglionic or postganglionic? The question you could ask is, where is the neuronal cell body from which that axon originates? If the answer is the cell body is in the peripheral nervous system living in a ganglia, then by definition it is a postganglionic fiber. Or if the answer is the neuron is residing in the central nervous system, spinal cord or brainstem, then the answer must be preganglionic fiber. So if you know where the neuron is from which the fiber is arising, the fiber in question, then you can answer the question correctly. So that's one way of looking at it. The highest control center for the autonomic system is the hypothalamus, which is in the center of your brain. So it is the hypothalamus that will be sending signals to this neuron here. So we're going to go hypothalamus. Okay, the neurons in the hypothalamus will talk to neurons here in the central nervous system, and then those neurons will then in turn send a fiber or axon that leaves the central nervous system to find its appropriate ganglia. Now, rest assured that the hypothalamus is not acting on its own. It has a lot of input from other neurons in the brain. The hypothalamus also can be affected by hormones and a lot of other biologically active molecules. So, again, a lot of influence on the hypothalamus. The hypothalamus then orchestrates the neurons in the central nervous system, and these neurons here are also either in the brainstem or the spinal cord. And they will then send their axons out, and then they will synapse onto a ganglia, and then those neurons will then talk to the end organ. Let's break down now the sympathetic and parasympathetic, and I'm going to be fairly general with you on this. As far as the sympathetic system is concerned, now, when I say sympathetic system, where you probably have heard of it in terms of fight or flight. So it is a system that gets you worked up. It is a system that increases your awareness. It gets ready for you to flee or to fight. My simple explanation of this, and this is what I use all the time explaining the sympathetic system, I call it the bear in the woods event. So you can imagine if you are walking in the mountains somewhere by yourself, alone, and you round a corner of a path, and you are face to face with a big old grizzly bear, eyeball to eyeball. And the grizzly bear is showing you his teeth. My guess is you are going to have an immediate physiological reaction. Your body will change. Your heart rate will go up. You will sweat profusely. You will breathe heavily. And of course, you will have a certain emotional valence associated with said events. Fear. So, what has happened is you processing the existential threat of the grizzly, you're going to have a neurological, physiological reaction to that. That is your sympathetic event. It's a bear in the woods event. That's what I call it. And so your body is getting ready to fight or flight. You're either going to flee from the bear or you're ready to fight the bear, depending upon how frisky you feel at the moment. But you're going to do one or the other. So why is your heart pounding? Well, it's pounding in anticipation of your skeletal muscles needing a lot of extra blood, glucose, oxygen. Why are you breathing heavily? Same thing. I'm going to need lots of oxygen. So whether I am running from the bear or fighting the bear, I'm going to need extra blood flow and extra oxygen. So why the sweating? Because in my effort to fight or flee, I'm going to be generating lots of heat, and I need to dissipate that heat. So again, the physiological response here has to do with preparing your body for intense physical activity. As far as the sympathetic system is concerned, you have a nucleus of neurons. Again, I'm using that word now, so you should know what that means. A nucleus of neurons meaning a collection of neurons somewhere within the central nervous system. And the one that is associated with the sympathetic system is called the intermediolateral cell column. And oftentimes you'll see it as the IML. This is actually a column. This nucleus is a column, and it lives in your spinal cord, from roughly level T1, T1 being thoracic 1. So you have spinal cord segments in your spinal cord, and they are roughly having to do with vertebra in your back. So you know that you have 12 thoracic vertebrae, and a thoracic vertebra is by definition attached to a rib, so you have 12 pairs of ribs. So your T1 is basically at the base of your neck, and you'll go down to T12, which is going to be about mid-back. So whenever your ribs run out, that's about where your thoracic vertebra runs out. And then, of course, caudal to your thoracic vertebra will be your lumbar vertebra. Going back up to your thoracic, rostral to your T1 vertebra will be your cervical vertebra. So you have numbered vertebra, and then you have numbered spinal cord segments that have something to do with the vertebra. They're not exactly lined up. In fact, the farther down you go in your spinal column, the less they are aligned, but they have something to do with each other. So that's close enough. So if I say I have a collection of neurons that runs from T1, that is thoracic level spinal cord 1, to L2, which is lumbar spinal cord segment L2, then that column of neurons are going to be the neurons that send their axons from the central nervous system to their external target. In this case, it would be a downstream ganglia. So we're talking about these guys right here for the sympathetics. These neurons here would be T1 through L2 in the spinal cord. Now, this is a bilateral structure, so you have a left one and you have a right one. And I have a cartoon on the next page that will help you with this. So those column of neurons, T1 through L2, are receiving input mostly from the hypothalamus. And of course, the hypothalamus is receiving lots of input from other systems. So they will come in, they will synapse, stimulate the IML. The IML will then send signals out, action potentials, to the neurons that leave the spinal cord and will talk to some other downstream ganglia. Here, you see. Now, I have some verbiage through here. It alludes to sympathetic chain ganglia, etc. That is not going to mean anything to you now. It will after I show you the drawing on the next page. So this paragraph, or the rest of this paragraph, will make sense or more sense to you when you see some cartoons. So I'm not going to burden you with that at present. The neurotransmitter that is used in the downstream ganglia, so now I'm talking about, let me clean this up a little bit. So I am talking about the neurotransmitter that's used here. And interestingly enough, whether it is the sympathetic system or the parasympathetic system, the same neurotransmitter is used. And the neurotransmitter that is used either in the sympathetic or the parasympathetic is acetylcholine. And we usually see this as ACH. Acetylcholine, very common neurotransmitter. Interestingly enough, it was the first neurotransmitter discovered back in the early 20th century. Oh, the 19-teens, somewhere along in there. There were two guys, scientists, last name Dale, and the other name is Lowy. they discovered acetylcholine and its function as a neurotransmitter. This was the first neurotransmitter. There was a long debate in science and medicine as to what was the nature of how electrical impulses were communicated from neuron to neuron or neuron to muscle. And the big debate was whether it was all electric or whether it involved a neurotransmitter or another molecule. And that debate raged for a fair while into the 20th century. Some people thought it was purely electrical. Others thought, no, you had to have an intermediate chemical or a molecule, which would be called a neurotransmitter, involved. So let me frame it this way. You know from high school or freshman biology that you have an axon coming out of a neuronal cell body, that the axon can have action potentials going down the axon. So how the signal is communicated from the beginning of the axon to the end of the axon, that's all electrical. So the question was, as one neuron talked to another neuron, was it somehow just a communication of that same electrical signal that just created an electrical signal in the downstream organ without any other chemical involved? Or was there release of a chemical that then in turn caused the next electrical impulse in the downstream nerve or neuron? That was the big debate. Is it electrical or is it chemical? Well, this was one of the first studies that suggested that it was probably involving another molecule, that it was not all electrical. So one of the first experiments on this, and it was done by Lowy, he was working with the vagus nerve, which you may already know and will know is a large nerve in the parasympathetic system. And its neurotransmitter, as we will about to see in a moment, is also acetylcholine. So they knew acetylcholine existed because Dale discovered its existence early on. And so what Lowy was doing was experimenting with the vagus nerve and a turtle heart. And he discovered that if you stimulate the vagus nerve, it slows the turtle heart down. And we'll find out in a bit why. So the vagus stimulation of the heart slows the heart rate down. So he did that experiment repeatedly, and again, stimulate the vagus nerve, going to the heart slows down, easy enough. So what he did was something rather clever. He took the fluid that was in the vicinity of the heart. Now the heart was taken from the turtle itself, so it's isolated, but still around some fluid so that it can maintain its metabolic integrity. It still can function for a little while at least. So and as you also know, the heart beats on its own. The heart does not need the central nervous system to beat. The heart has its own auto rhythm, and so the vagus nerve can influence the heart rate, but it certainly does not cause the heart rate. So what he did, he had two turtle hearts, again trying to figure out, is this purely electrical or is it chemical? So he stimulated the heart of the turtle, again it was removed, and then took the fluid around that heart that had slowed up in response to the vagus nerve, and then applied that fluid to another beating turtle heart that was not innervated by the vagus nerve. And when he did it, that heart also slowed down. So that was the first hint that something had to have been released, in this case acetylcholine, on the heart, and that you could take the acetylcholine and apply it to another beating turtle heart, and it would in fact slow it down. So that was suggestive that the nature of neuronal signaling had to do with chemicals as opposed to pure electrical impulses. So that was big stuff in the day, and Dale and Lowy actually won a Nobel Prize for that research. So a little bit of history on acetylcholine. Okay. Acetylcholine is not the only neurotransmitter involved. We're going to find out with other systems that norepinephrine and dopamine are also involved in this. So I think probably the best thing is to go to a diagram so that we can clarify this even further. And I'm going to draw your attention to these diagrams here to help us out. So again, we saw this earlier. That's my somatic. Okay, this is going to be, again, skeletal muscle. And what we find is acetylcholine is the neurotransmitter that these motor neurons, these are motor neurons, acetylcholine is actually released onto skeletal muscle for causing skeletal muscles to contract. So here we have the autonomics broken down to a parasympathetic and sympathetic. Both of them involve a ganglia. Ganglia, ganglia. And as I mentioned earlier, both of them release acetylcholine on that ganglia. Now, what happens is, now again, this is my pre-ganglionic sympathetic fiber. Post, well, I guess the P and that doesn't really work, does it? So let's just try this. Be a little more elaborate. This is my pre-ganglionic sympathetic fiber, and this would be my post-ganglionic sympathetic fiber. Okay, up here would be my pre-ganglionic parasympathetic fiber and my post-ganglionic parasympathetic fiber. So both the autonomics, para and sympathetic, both involve acetylcholine on the ganglia here, right? But when you go downstream, if we're talking the sympathetic, as far as the effector organ is concerned, one of the major ones is actually norepinephrine. It's not the only one, but it is the major neurotransmitter being used. Whereas on the parasympathetic, the acetylcholine is being released here on the effector organ as well. So while the neurotransmitters are the same in the ganglia for both sympathetic and parasympathetic, the neurotransmitter being used from the postganglionic fiber is different. In the parasympathetics, it's acetylcholine, and in sympathetics, it's norepinephrine. So this is the acetylcholine here that they were worried about or was looking at in terms of what was released on the turtle heart. Because if we're talking parasympathetics, which would be the vagus, of course, and then that case, the effector organ was the turtle heart. Okay, just to tie back our earlier illustration. Okay. Again, we'll return to parasympathetics in a moment. I do also want to share an aspect of the sympathetic system regarding the adrenal medulla. You know we have an adrenal gland, and its location is actually in the name, renal, okay, having to do with kidney. So your adrenal glands are sitting on the top of your kidneys, just happens to be there. And as you recall, perhaps from an earlier course, you have two different parts of the adrenal gland. You have the interior part of the adrenal gland, and that is called the adrenal medulla. And then you have an outer region, and that is called the adrenal cortex. And they're really two different glands that happen to be in the same place. Two completely different functions. So as far as the autonomics is concerned, we are focusing in on the adrenal medulla, the interior region. Of course, medulla means interior region. So it is within the medulla that you have neurons. And again, since we're talking sympathetic, there is my IML, my intermediate lateral cell column, T1 through L2. And so these neurons are here. Those neurons either go to a ganglia, or some of these neurons will go to the adrenal medulla. They will release acetylcholine. The nerves or the cells in the adrenal medulla, though, are not neurons. They're not ganglia like we have up here. So let me cartoon it out for you. Imagine I'm going to take these guys here, and they're going to be releasing their acetylcholine. The neurons in the medulla are just regular cells. And I have running through there, of course, blood vessels. I'm just going to go with that blood vessels there. The acetylcholine will bind to receptors on these cells and will stimulate these cells to release their contents, and their contents are going to be mostly epinephrine. The epinephrine is released, picked up by blood vessels, and distributed throughout the rest of the body. Those epinephrine molecules will then bind to receptors throughout the rest of the body, causing enhancement of function. Again, it depends upon the receptor, depends upon the organ. So you have two things kind of going on when you have a bear in the woods event. One is the system itself, the IML, and then the effector organ here. But the other is it's affecting your adrenal medulla. So I'm getting large amounts of norepinephrine on various end organs, but the adrenal medulla is releasing a hormone, epinephrine, and it is being carried throughout the body, also influencing. So you get kind of a double barrel shot of stimulation, so to speak. And the combinations of these allows you then to function in a way that is greatly enhanced. We've all heard the story of little old lady, grandma, 80 years old, walking her little dog, Fifi. And then you have a 4x4 rounding a corner, flips, crashes, and lands on little grandma's Fifi, little puppy. So now puppy is trapped under the 4x4 that is flipped. And grandma tosses her walker aside, runs to the 4x4, grabs it by the back bumper, lifts it up, and grabs her little precious Phoebe. And the dog is saved. How is that possible? Because in the moment of sympathetic stimulation through a variety of cellular processes, many of your functions are supercharged. Your muscles can do things that they normally could not do because of this surge of stimulating bioactive molecules called epinephrine and norepinephrine. To be sure, grandma will be quite sore the following day, but that is the power of sympathetic stimulation. complements of norepinephrine and epinephrine. So I think I will end this here, and we'll pick back up on this diagram in the next block. I want to spend a little bit of time on this diagram. As you look at it, it may be a little disorienting, but I'm hoping to, when I'm done with you, that it will make sense. What you're looking at is a cartoon of a horizontal slice through a spinal cord. Now, mind you, this is only part of the spinal cord. Remember, we have, I could fill it in here, you see. So again, just making sure you understand your orientation. So the spinal cord is made of gray matter. It's that little butterfly thing in the middle, and that's where most of your neuronal cell bodies are. And then it is going to be surrounded by white matter. Okay, so again, white matter and gray matter. The white matter is going to be dominantly axons. The reason it is white is because many of those axons are myelinated, so that gives it a whitish texture. When I say the gray is where the neurons are, I have to be careful. That's where the neuronal cell bodies are, because the neurons are certainly in the white matter because axons are part of a neuron. So I'm making a distinction between neuronal cell bodies and the axons. And what I'm telling you is the white matter is just mostly axons. Gray matter is both axons and neuronal cell bodies. And of course, it's a horizontal cut. So as the cartoon goes here, though, let us orient ourselves that this is the dorsal part of the spinal cord. And I could also say that this is the posterior part of the spinal cord. Here is the ventral, and I could also say this is the anterior part of the spinal cord. So posterior equals dorsal, ventral equals anterior. When you are speaking in science language, the more common vocabulary is dorsal and ventral. The reason that is the case because the world of bench experimental scientists have to do, or historically had to do, with working with four-legged critters, mice and rats and such. And so they are, of course, four-legged. So their spinal cord, spinal column is parallel with the floor. And so ventral is the belly, dorsal is the top, and that's why that vocabulary is employed. Of course, humans were bipedal, so we're upright. So our spinal column is perpendicular to the floor. So since we're upright, it's more appropriate to speak of an anterior and a posterior. But if we got on all fours, our back, our posterior is dorsal, and our underside or belly is ventral. So any of the structures I refer to here, if I say dorsal, I could have easily said posterior. If I say ventral, I could have easily said anterior. Either vocabulary works. So coming out of the spinal cord, we have nerve roots. And these roots, I have a ventral root. Remember, I could have also said anterior root, so ventral root. And then I have a dorsal root. And I could have said posterior root and been equally correct. I will make a generalization. While this is not absolutely true, it's mostly true, which is the ventral roots are associated with motor function. The dorsal roots are associated with sensory function. So with sensory function, of course, you have the signals going into the central nervous system coming from some peripheral organ, whether it be tactile for skin, or whether it's taste on the tongue, or whatever. Fill in your own sensory, visual, auditory. Those signals are coming from some other, peripheral part of the body going back into the central nervous system to be appropriately processed. Whereas motor is leaving the central nervous system and going out to an end organ. So the signaling for the dorsal, all the signaling is going into the central nervous system. For motor, signaling is going out of the central nervous system. Now this little bleb here, that is called the dorsal root ganglia. And what did we say a ganglia was? A collection of neuronal cell bodies outside the central nervous system. This dorsal root ganglia is a sensory ganglia. A ganglia could either be sensory or motor. So all sensory ganglia are made of what is called pseudo-unipolar neurons. Pseudo-unipolar neurons, and they look something like this. That's the cartoon version. So these neurons here, of course, are going the rest of your body. They're coming in, and they will come in on the dorsal root, and they will then make a synapse onto some other neuron in the gray matter, and then that neuron will then send this signal back up to the brainstem and higher brain centers. That's as detailed as I'm going to go on that, because I'm really not wanting to talk about the sensory system at this moment. I do want to focus, though, on the motor, in particular the autonomics. So before I go autonomics, let me make one last point here. The somatic motor, the somatic nervous system. Remember, that is your motor voluntary system. So I'm going to have a neuron sitting in the ventral horn. By the way, this is the ventral horn of the gray matter, and this is the dorsal horn of the gray matter. And you guessed it, I could have called it the anterior horn of the gray matter or the posterior horn of the gray matter. So the sensory neurons tend to be in the dorsal horn of the gray matter, whereas the motor neurons tend to be in the ventral horn of the gray matter. So if I'm talking voluntary movement, I'm going to have motor neurons living in the ventral horn of the gray matter, and they are going to send their axons out into the various branches and getting to the body. Okay, so they're heading out. Okay, again, this is for voluntary. And by the way, the ventral root and the dorsal root, they will join together. And it's a very short segment, but they will join together, and then they are going to form what is called the spinal nerve. The spinal nerve is short. It will then bifurcate into two branches. And my first branch is here. Different color. This branch here, this is my ventral primary ramus. And then over here is going to be my dorsal primary ramus. And you guessed it. I could have said anterior, posterior, and been just as right. Ramus comes from the word to ramify. And in medical speak, to ramify means to branch. So ramifications means branching. So a ramus is a branch. The primary rami, plural, rami, will then lead to secondary, tertiary, quaternary, quaternary, so forth, so on. So you're just going to have lots of branching downstream as these nerves get more and more branchy. They'll ramify. They'll branch. Okay. But the spinal nerve itself, again, isn't very long, and then you immediately have a bifurcation to the primary rami, dorsal, and ventral. Okay. So now let's go back to focusing on the sympathetics. And I don't like where this is drawn. The sympathetic, the IML should be right there. That's a better place for it to be. So we just kind of bump it there. Now again, a reminder, this is my cell column. So I got one over here and I got one over here. And these neurons, again, are going to be present in the spinal cord segments T1 through L2. So these neurons are not in all of the spinal cord. They're only in that segment, T1, T2, T3, all the way down to L2. And they live at the lateral little horn here, little hook here, at the lateral side of the gray matter. Now, the neurons that live here, these are the ones that are receiving signals from the hypothalamus. So they're coming down, sending signals to the hypothalamus, from the hypothalamus, stimulating those neurons. These neurons will then leave the spinal cord, got the little red here, they'll travel in the ventral root along with the regular motor neurons that are involving voluntary motor function. But these fibers here take a curious path. So they're going to go here, here, and oh my, look what happens. So now they are going a different direction relative to my voluntary motor neurons that were in the green here. So now we have to introduce another structure. And this other structure is another ganglia, and there he is right there. This ganglia is called a sympathetic chain ganglia. There he is. That's one of them. Now note the word chain. And that is because you have another one here, and then another one here, you got another one here, and so forth, so on. So I'm only showing you one. But these little blebs, these little Y things, they go up and down your body cavity. This is a bilateral structure, so you have two sympathetic chain ganglia. The most rostral aspect of your chain ganglia is actually at the base of your skull, buried in the soft tissue of your neck. You got a right one and a left one. That chain goes all the way through your neck, goes into your thoracic cage, leaves your thoracic cage, goes into your abdomen, all the way down to your pelvic bowl. So your sympathetic chain ganglia is roughly 3 feet in length. Now, I can talk a blue streak. You're not going to get it. So what I need for you to do is to get on the internet and Google sympathetic chain ganglia and look at some diagrams. You need to see it. Once you see it, then what I'm about to tell you makes a lot more sense. But please look it up. Get your eyes on a really good diagram, and then this will make more sense to you. Okay. Let's go back to our chain ganglia. So we now have this preganglionic sympathetic fiber. Can I use that word now? Preganglionic sympathetic fiber. It is going to leave the ventral ramus and will travel in another nerve. And this nerve here is called a white ramus communicans. This one here. This is my white ramus communicans. my white ramus communicans contains preganglionic sympathetic fibers. The sympathetic chain ganglia is called ganglia for a reason. There are neurons there. So what will happen is that this preganglionic fiber will synapse release acetylcholine onto the neuron that lives in the chain ganglia, and it will then send its fibers out, and in the scheme here, it's a line and a dot and a line and a dot. So dot, line, dot, line, dot, line. And note that those axons are returning to the spinal nerve / ventral primary ramus. This nerve here is called the gray ramus communicans. Gray, and I'm going to go RM for ramus communicans. It is called a gray ramus communicans because it looks gray. The white ramus communican is called that because it looks white. And that is because the neurons traveling in the white ramus communicans are myelinated, so it's white. The neurons in the gray ramus communicans are unmyelinated, so it looks darker or looks grayish. These fibers will then re-enter, and again, I'm going to correct the drawing here. Many of these fibers are going through the ventral primary ramus, but some of these fibers also go through the dorsal primary ramus as well. And they're going to be heading to the periphery of my body. They're going to be communicating to my skin. They're going to be communicating to my skeletal muscles. Where these fibers do not go is to any visceral organ in my body cavity. These only go to parts of my body that is not my body cavity. Thoracic, abdominal, or pelvic. There are other pathways that involve this scheme that I'll talk about in a moment. So when you have a Bear in the Woods event, sweating will be from these fibers that I just showed you. One of the other things that happens when you are frightened is you get goosebumps. The erector pili muscles are contracting, raising your follicles. Again, this is from this system here, you see. So what these fibers are not involved in, though, would be visceral organs. Okay? So let me draw you two other patterns so that we can complete our story of the sympathetic system. And here we go. Let's go back to our preganglionic fibers of my sympathetic system. So I'm going to go back to red. So I said earlier that these will synapse onto neurons in the chain ganglia. Many of them do. Some of them don't. Some of them will not synapse, but instead will go right through the ganglia and will find another ganglia somewhere else in the body. So this is something you have to be careful of. So the question is that a preganglionic or a postganglionic sympathetic fiber? The impulse is to say, well, it's postganglionic. Why do you say that? Well, because it is coming from the chain ganglia. That's true, but you have to go back to the question that I asked you to ask, which is, where does this neuronal cell body live? Well, it lives in the IML. So this is still a preganglionic fiber. Sympathetic fiber. Because its neuronal cell body is still up here. It is making its way to its own ganglia. So there are other sympathetic ganglia that are in the body in addition to this chain ganglia. These ganglia here live in your body below the diaphragm. Below the diaphragm, as in your abdominal or pelvic areas. You have many of these ganglia here. All have different names that you'll learn in gross anatomy, by the way. I'm not worried about it learning here. So you have sympathetic ganglia below the diaphragm that are spread throughout your abdomen and pelvic area. They are receiving the preganglionic sympathetic fibers. They will then be synapsed upon. They will receive their acetylcholine. Then those ganglia will send their fibers out to whatever end organ they're going to affect. And of course, because it is sympathetic, the dominant neurotransmitter will be norepinephrine. And of course, acetylcholine is released here as well. So any sympathetic event or effect of any organ below the diaphragm is going to be this theme here. So now we have one other scenario. Let's take care of it. And there we go, that funky color. The population of neurons in the sympathetic chain ganglia, I got the green ones there. They're the ones that are going to be going to your skin and skeletal muscles on your extremities. I have another population in here as well. They will also receive acetylcholine from the axons from the IML. They'll synapse here, and instead of returning to the spinal nerve via the gray ramus communicans, they're also going to leave and form their own nerve, and they will then synapse on the end organ. These are post-ganglionic sympathetic fibers, and they'll be releasing their norepinephrine on their end organs. This is the pattern by which the organs in my thoracic cage are innervated, and so the biggies are going to be your heart and your lungs. So any sympathetic nerve that you find in your thoracic cage are by definition postganglionic. Their neurons live in the chain ganglia. Whereas neurons that are running in your abdomen, those are likely to be preganglionic fibers, and they're en route to find their ganglia on which they will synapse, and then they will then emit their postganglionic sympathetics. These ganglia tend to be fairly near where their effector organ is. So now what I've shown you are three different scenarios for your sympathetics. One theme has to do with my skin and skeletal muscles, or was everything outside my body cavity. And then within the body cavity, I have one theme for above my diaphragm, and then I have another scheme or theme for below my diaphragm. So that's kind of a summation of how all of this works. So we will end this block here and pick up next time. And again, don't forget, go on the internet and look up and get your eyes on a diagram of your sympathetic chain ganglia. That will make all the difference in the world for you. See you next time. When the sympathetic system is activated, it is not the case that all aspects of the sympathetics are activated. So it's not all or none. You can have some aspects of the sympathetic system are activated and some are not. So let's just go through some of the effects of the sympathetic stimulation, again with the understanding that if you increase the sympathetic tone or increase the sympathetic signal, it's not the case that all of these things happen. So some of the effects, you would have an increase in heart rate. We've already spoken of that. It's called tachycardia. A widening of the pupils. So the iris would contract. You'd have contraction of the radial smooth muscles. Let me tarry on that a little bit. You already know that pupillar size is an autonomic response, and that you have antagonistic autonomic input to the smooth muscles that constitute the iris. The sympathetic system will cause the pupil to grow larger. The parasympathetic system will cause the pupil to get smaller. You also know that it is light responsive. So you're aware if you shine a light into a pupil that both the pupils will contract. If you darken the environment, then the pupils will get larger. The iris is made of a series of smooth muscles, and you have two different arrangements of the smooth muscles, so I'm just going to cartoon it out for you here. Some aspect of the iris, it's basically a functional sphincter. So you have these circular arrangements of smooth muscles, as I'm trying to draw there. So such that when those smooth muscles contract like a sphincter, it will get smaller, and that will cause your pupil to, again, of course, get smaller, and that would be in response to light. So the autonomic system that innervates these smooth muscles are going to be your parasympathetics. You have another set of smooth muscles that are arranged more of a radio-like configuration, and you can guess what's going to happen here. And those are going to be innervated by your sympathetic system, such that when they are contracted, they will draw the pupil open and it becomes larger. We're going to see this theme over and over again. It's not that the sympathetics and the parasympathetics send no signal and then all of a sudden they send some signal. For most of the systems, you have a baseline autonomic tone. So even at present, you listening to this lecture, you're having a certain level of sympathetic input to your iris and a certain level of parasympathetic input to your iris. So you have these two sets of muscles, the more radially arranged and the more concentric circles, or the sphincter-like arrangement, are both being contracted at the same time, but they are antagonistic to each other. So for pupils to alter their diameter, all you have to do is just stimulate one part of the autonomics a little more than the other, or you could get the same effect by dialing back the tone of one over the other, same difference, and you would then alter the size of the pupil. So like with other systems we're going to see, you always have some degree typically of some sort of parasympathetic tone if the parasympathetics innervate a particular organ, and a sympathetic tone. It also makes sense from an engineering perspective in the sense of if you have a baseline tone over a particular system, then do you not have two ways to go? You can either back off the tone or you can increase the tone. If your default situation is just simply none, then you only have one way to go, which is turning it on. So we'll see this over and over again where you have a mid-level tone or some degree of tone such that it gives you two options. You can either dial the tone further back or you can increase the tone to get whatever desired effect that you want. Other effects of the sympathetic stimulation reduces blood flow to peripheral areas of the body, such as skin, fingers, toes, and such. And again, think about Bear in the Woods. Your blood is better spent for muscles and for heart and for lung. So your body naturally will reduce blood flow to those areas of the body that have less priority for blood flow during the Bear in the Woods event. So it does kind of make sense. You have an increase in blood flow to the muscles, talking skeletal muscles there. You'll have body hair erection. Those are erector pili muscles that will give you the goosebumps. Another side effect of, or the effect of a sympathetic stimulation is going to be inhibiting salivary glands and also inhibiting GI contractions, digestion of food and such. There's a connection between these two, the salivatory glands and the digestion. That's all part of the same system of the GI tract. Of course, your salivatory glands creating enzymes that begin to digest food in your mouth. So the theme during a sympathetic stimulation is that it will inhibit the GI function. So your mouth would grow dry, and if it's a powerful sympathetic system, it can grow dry pretty quickly. Also, your gut motility will decrease because if you're confronted with the grizzly and he is showing his pearly whites to you and he is looking at you as his next happy meal, you're more concerned about not being his meal compared to digesting the last meal you just ate. So digestion of food is not a priority during the acute phase of a sympathetic stimulation. Sympathetics will also relax bronchial muscles of the lungs. So here's a situation where the sympathetics actually relax muscles. And again, that's going to be a function of the type of receptors that are on the smooth muscles that are responding to, say, the epinephrine, or the norepinephrine, I should say. Again, it makes sense. You're wanting to relax the bronchial muscles to increase the diameter of the air passages, such as to facilitate movement of air in and out of the lungs. A couple other things. The sympathetic system is very much involved in ejaculation during sexual endeavors, on the male side, of course. It also inhibits micturition, and micturition is a fancy way of saying to void one's bladder. And then it also inhibits defecation as well. Now, you might question yourself in terms of the last two. The sympathetic does inhibit defecation or the voiding of the contents of the rectum. That event is complicated. It is an interface of parasympathetic and sympathetics because, again, as the common question is, you're so frightened that you lose your control of your bowels, and also you pee in your pants because you're so frightened. That can happen. So I'm not implying that that can't happen. I'm just saying in normal tones of sympathetics as we're going day to day, that the sympathetics can and does inhibit those two events, voiding the bladder and voiding the rectum. But under certain circumstances, it can go the other way. I know when I was research active, I did a lot of surgery on rats, testing rats, and one of the fear responses of rats is to defecate and to void the bladder. So it can be involved in that. So it gets a little complicated in terms of the interplay of the autonomics having to do with those particular bodily functions. So I'm just going to leave it at that and just understand that these are certainly not absolutes. But again, my major point here is that the sympathetic system is not some monolithic thing. You either turn everything on or everything off. You can have sympathetics involved in different things, but not necessarily involved in other things that the sympathetics could be involved in. So, example, I mean, you don't get goosebumps if you shine a light into somebody's eyes. You know, I said that the opposite. When you put them in darkness, you know, that sympathetics is going to cause the pupils to grow larger. Again, the light response to darkness, such as the pupils are getting larger, doesn't mean you get goosebumps at the same time. So the sympathetics can go to different organ systems in different degrees. So it's not a monolithic type of thing. Let's turn our attention to the parasympathetics. We had said earlier that the sympathetics, the neuronal cell bodies that give rise to axons that actually will go out and leave the central nervous system, they all live in two columns in the spinal cord, the intermedial lateral cell column from roughly T1 to level L2. Again, you have two of them. And of course, they're going to interact with the sympathetic chain ganglia, and I hope you've looked that up so you can get your eyes on it. The parasympathetics are a little different. You have two populations of neurons in the central nervous system that service the parasympathetic, and they're the analog to the neurons that are T1 through L2. But they're at different places. So some of these parasympathetic neurons in the central nervous system are associated with cranial nerves. And I hope you have at least a vague notion of what cranial nerves are. You have 12 pairs of cranial nerves, named cranial nerve 1 through cranial nerve 12. The cranial nerves mostly are involved in function of the head, the face, although you have some cranial nerves that influence systems beyond that. I'm thinking of cranial nerve 11, the accessory. It's going to be contracting your sternocleidomastoid muscle and your trapezius muscles. I'm thinking of the vagus nerve. That's cranial nerve 10. It certainly innervates, your larynx. It innervates your visceral organs down to parts of your digestive tract. So some of the neurons live in the brainstem that are associated with the cranial nerves. You have 4 cranial nerves that have parasympathetic function. And those cranial nerves are cranial nerve 3, that's your ocular motor, so that's going to be involved in your eye movement. You have cranial nerve 7, that is going to be your facial nerve, and that is involved in moving the muscles of your face, but it also has a parasympathetic function. You have cranial nerve 9. It's going to be your glossopharyngeal nerve. Okay, that nerve is involved in sensory of the throat, but also it has other parasympathetic functions. And then finally, the one we just mentioned, cranial nerve 10, which is your vagus. And again, it has many functions, one of which is parasympathetics. Now, what I'm not suggesting to you is that these 4 cranial nerves, that's all they do. They don't. All I'm saying is, as part of what these 4 cranial nerves do, is involved in a parasympathetic function. The vagus nerve, powerful parasympathetic function to your heart, your lungs, your digestive tract, etc. But the vagus nerve is also very much involved in innervating the skeletal muscles associated with your larynx that allows you to speak. So a cranial nerve can have multiple modalities. There can be a non-autonomic aspect to it, and there can be an autonomic aspect to it. And if we're talking about cranial nerves, then we're talking about some of them, these 4, have or can have or do have a parasympathetic function. But that is not to imply that's all they do. So the neuronal cell bodies that service these 4 live in the brainstem, which makes sense, of course. You then have another population of parasympathetic neurons, and again, they're the analog of the sympathetics T1 through L2, and they live in the sacral spinal cord, S2, S3, S4. Those neuronal cell bodies are going to be responsible for all parasympathetic function in the lowest part of your digestion tract and also organs of the pelvis. We're talking bladder, we're talking rectum, we're talking genitals. I had mentioned earlier that the vagus innervates a large portion of your GI tract. The vagus will innervate your GI tract all the way down to the splenic flexure of your colon. You recall from high school anatomy that on the right side of your body you have an ascending colon, and then about the level of where your ribs end on the front part of your body, you have a transverse colon. It goes to the left side, and then the colon then takes another 90-degree turn on your left side and goes down. And then that descending colon will then transition into the sigmoid colon and then eventually leading to the rectum. The vagus nerve will innervate, supply parasympathetic signaling throughout the GI tract down to the splenic flexure. The splenic flexure, fancy medical word for bend. Spleen, if you know your anatomy, your spleen is on the left side of your body cavity. So the splenic flexure of the colon is that 90-degree turn from the transverse colon to the descending colon, and the spleen is actually right behind it. So splenic flexure. So what about the descending colon, sigmoid colon, rectum? The parasympathetic input for those are going to be picked up by these parasympathetic neurons that live S1, S2, S3. So that's what they service, in addition, of course, to the pelvic organs, genitals, and such. A couple other differences. I had mentioned earlier that while both autonomic systems, parasympathetic and sympathetic, the neurotransmitter is going to be acetylcholine. The end organ neurotransmitter, as I said, for sympathetics is going to be mostly norepinephrine. The endpoint neurotransmitter or end organ neurotransmitter for the parasympathetics is going to be, again, acetylcholine. So, again, a slight difference regarding the arrangement of the neurotransmitters. So, with the parasympathetics, it's acetylcholine and acetylcholine. Other types of differences. While the sympathetics, again, the neurons leaving the central nervous system will find another ganglia. Again, the large ganglia, that sympathetic chain, but you also have other named ganglia we'll show in a minute. You have parasympathetic ganglia as well. So these nerves will leave the central nervous system, whether they're from the sacral or whether they're from the brainstem, will just leave the central nervous system. They are preganglionic parasympathetic fibers, and they're going to go out and they're going to find their parasympathetic ganglia. Now, what's different about the parasympathetics is that those ganglia tend to be very small, and there's a lot of them. And those ganglia tend to be very, very close to their end organ. So some of these ganglia may be 3 and 4 neurons, and that's it. But you have a lot of them. If you have taken histology or GI physiology, then you've probably run across neurons that are embedded between layers of the GI tract. Auerbach's plexuses is one of them. Again, what you're looking at there is just parasympathetic ganglia. But they're everywhere. This is part of the enteric system, which is the nervous system of the gut. Again, that is parasympathetic, and it is being talked to by the vagus nerve and being talked to, if you're in the lower part of the gut, by the parasympathetic neurons living in the sacral area. The neurons, brainstem or the sacral, they are also, like the sympathetics, under the control of the hypothalamus. And of course, the hypothalamus is gathering a lot of information from other systems in your body. So some common parasympathetic effects. Some of these tend to be antagonistic to the sympathetics, and then some not. So the heart rate, the sympathetics increase heart rate. The parasympathetics will decrease heart rate. And of course, if you're talking heart, you're talking vagus. So you have a vagal tone going to your heart. And I said earlier, the heart itself is an auto function. It doesn't depend on the nervous system to beat, but the nervous system certainly can influence its nature of its beating. So I have a baseline level of vagal tone to the heart, but I also have a baseline level of sympathetic tone to the heart. And if I were to just increase the vagal tone and not change anything else, it would be a slowing of the heart. Or if I backed off the vagal, my heart would increase if I don't change my sympathetics. So again, lots of ways of controlling that. As I'd mentioned earlier, the constriction of the pupil. So you can see the diagram up here in terms of the sphincter component of that. Stimulation of the salivatory glands and the GI tract. Again, the parasympathetic is very much involved in the digestion of food. So if you're hungry or you have some type of cue in the environment, maybe it's a smell of baking bread in the oven and you're hungry, or maybe it's a smell of, seasoned steak out of the oven, or maybe you have a apple cobbler pie that's filling the room and you're hungry, your mouth starts watering, you get really hungry, your peristaltic reflexes start churning in your stomach. All of that is complements of the vagus nerve and your parasympathetics preparing the body for a very fine meal. The parasympathetics can contract the bronchial muscles in the lung, again, antagonistic to the sympathetics. You may ask, why would I ever want to contract and make smaller the air passages in the lung? There are very good reasons for that, and we will explore those reasons when we do pulmonary physiology. The parasympathetics will stimulate micturition, defecation. And again, sort of antagonistic, but again, I've got some asterisks by those because, again, with fear response, it is not, put it this way, it's not as simple as the parasympathetic sympathetic antagonism of the pupil diameter. It's a complex weave. And then the parasympathetics are very much involved in penile clitoral erections because they have innervation of erectile tissue in genitals. And I believe you're all aware that, and let me just kind of pause on this just for a second. Almost all of your blood vessels in your body have rich sympathetic input, with the exception of blood vessels in the central nervous system. They don't have much in terms of sympathetic input, but once you're outside the central nervous system, you have a significant sympathetic tone to all blood vessels. You do not have matching parasympathetic tone to all blood vessels. In fact, the vast majority of the blood vessels in your body have no measurable parasympathetic input at all, with a few exceptions. And the exceptions are, one of them is going to be your erectile tissue of genitals. And so we all understand that with the engorgement of blood during sexual activity, that part of that, that is a result of the parasympathetic stimulation of those blood vessels that causes those blood vessels to relax, allowing more blood to go into the erectile tissue than leaving, and then you end up with engorgement of blood, and there you go. You do also have some parasympathetic input to blood vessels of the coronary arteries as well, and you could find a few in blood vessels of the GI tract and such, so there is some. The physiological effect of that, people will argue, most people argue it's not a major physiological effect, but they're there, and so need to acknowledge that. Okay, so we'll just break at this point and pick back up on the next page. I would like to turn our attention to receptors. And what I'm about to tell you is a fundamental concept. When you have neurons talking to other neurons, or you have neurons talking to muscles, whether it be cardiac, whether it be smooth or skeletal, what the particular neurotransmitter does depends strictly upon the receptor. You have a myriad of neurotransmitters. Each neurotransmitter, some neurotransmitters are excitatory, which means they're going to stimulate the downstream organ. Some neurotransmitters are inhibitory, which means they would inhibit downstream organs. But whether they are excitatory or whether they are inhibitory or whatever the biological effect is of that neurotransmitter, it is strictly determined by the receptor. I will say this many times throughout this course. If you let me design the receptor, I can make a neurotransmitter do anything I want. It is all about the receptor. And we're going to see this throughout the course. So up to this point, we have spoken of two basic neurotransmitters, norepinephrine, or epinephrine, similar, and the other is acetylcholine. So let's talk about some of the receptors, and also bear in mind that you have various subtypes of receptors and you have subtypes of subtypes. They're all various isoforms of each other that can have different effects on the cells. So we'll start off with receptors that are going to be binding to acetylcholine. That's my ACH. So again, the ACH, that's going to be my ligand, or going to be my neurotransmitter. Neurotransmitters are released, and then they will bind to receptors on the cell membrane that they've released upon. So as far as acetylcholine is concerned, and we would call this cholinergic. So cholinergic receptors or cholinergic neurotransmitter, acetylcholine would be one of them. So you have two basic types of acetylcholine receptors. One are nicotinic receptors. The second is going to be the muscarinic receptors. Now, I will freely tell you there are subtypes of nicotinic receptors and there are subtypes of muscarinic receptors. And usually, like muscarinic, you'll have an M1 or an M2 or an M3 and so forth, so on. So at this point, I'm just wanting to be a little more simple. So the nicotinic receptors are what we call inotropic. An inotropic receptor is a receptor such that when the neurotransmitter or the ligand binds to it, it is a channel. It is a channel that will either open or close depending upon the receptor, and will either allow or disallow things to go through that channel inside to out or outside the end of the cell. So nicotinic receptors are inotropic, so that when acetylcholine binds to them, that allows then sodium, which you have a lot of sodium on the outside of the cell, a little bit of sodium on the inside of the cell. So sodium will go down its concentration gradient and also go down its electrical gradient, because inside the cell is negative and sodium is positive. So the opening up of nicotinic receptors in response to acetylcholine allows sodium to go in. This type of nicotinic receptor is found on all postganglionic neurons. So you'll recall that acetylcholine was the receptor of choice for both your sympathetic and your parasympathetics when those preganglionic fibers would then in turn bind to receptors in the ganglia. Those receptors in those ganglia, whether it's a parasympathetic ganglia or a sympathetic ganglia, are nicotinic receptors. They will allow sodium to go in, which will in turn effect an action potential in those neurons, and then those action potentials will travel through the postganglionic fiber. Nicotinic receptors are also the receptors you find on neuromuscular end plates. So when a motor neuron innervates a skeletal muscle, it will release acetylcholine. Acetylcholine will then bind to nicotinic receptors that are embedded in the cell membrane of the muscle fibers. And when the acetylcholine binds, it allows sodium to rush in to the skeletal muscle fiber, and that can then in turn effect an action potential on the muscle, which would result in a muscle twitch. The nicotinic receptor is named for nicotine. Nicotine binds to nicotinic receptors. So when they first were working out this receptor, they found this receptor that nicotine binds to, and it turns out this is the receptor we're talking about, so they call it nicotinic receptors. So nicotine binds to them. The other kind of receptor you have is muscarinic. Again, many subtypes. A muscarinic receptor is a metabotropic receptor. So a metabotropic receptor is a receptor that is not a channel, but it will bind to neurotransmitters. But when the neurotransmitters bind to the metabotropic receptor, it will alter cell biology within the cell. Usually these are associated with G proteins. And so if we're talking about receptors associated with G proteins, and we're talking about calcium, calmodulin, IP3, PIP, DAG, all the other things, it's calcium, all the things that we're talking about that could be involved in second messenger systems, phosphorylating, desphosphorylating. All of that chain reaction occurs when an acetylcholine binds to a muscarinic receptor. So a muscarinic receptor, metabotropic, means it's not a channel, but rather that it is going to alter the cell biology of the cell that that receptor is on. They're called muscarinic because certain chemicals found in mushrooms that have a muscarinic name attached to it will bind to these receptors. So this literally, you have muscarinic type of compounds in certain types of mushrooms, and this is where this came from in terms of its name. You find them on the membrane of the effector tissue that is innervated by postganglionic neurons. Okay, so be careful and don't cross this up. Up here, nicotinic on all postganglionic neurons. So let me draw this out for you, so I don't want you to get confused. So if this is the neuron in the central nervous system, okay, it's going to leave the central nervous system. And then I have a neuron here, and then a neuron, and that's my effector organ. I'm going to say EO. So here, these little red guys, those are nicotinic receptors, okay? So they're found on postganglionic neurons. So that's your postganglionic neuron right there. Whereas for this, it says found on the membrane of the effector tissue. So we're going to say that this in organ has these little muscarinic receptors. Okay, so this is on the membrane of the effector tissue, so that's my effector organ or effector tissue there. So make sure that language is clear to you. Okay, so we'll go back to here. Muscarinic receptors, metabotropic, found in membranes of effector tissue that is innervated by postganglionic neurons of the parasympathetic and sympathetic as well. Again, I'm going to talk about that in a second, because you say, wait a minute, I thought that the sympathetics only dealt with norepinephrine. They do, but a cartoon's going to come up and show you that sometimes acetylcholine is released by that postganglionic sympathetic neuron. That's the minority, but sometimes that happens as well. And that'll come about in a cartoon I'll show you in a page or two. Atropine is a muscarinic antagonist, and so make sure we're understanding that word there. So when we're talking about the activity of a biological molecule, we can talk about antagonism. And so if a molecule is antagonistic, then that means it does the opposite of what the normal neurotransmitter would do. If a molecule is an agonist, that means it does what the normal molecule would normally do. So we'll want to get comfortable with antagonism and agonism. So atropine, biological active molecule, is a muscarinic antagonist, which means it is going to shut down or inhibit normal activity of acetylcholine. You use atropine all the time in surgical situations. Typically, you'll give atropine to a patient in surgery to inhibit the secretions of the salivatory and bronchial secretions. And again, remember that the acetylcholine stimulates those things. So you'll have reasons to dial back the digestion, juices, etc. During surgery for a myriad of reasons. I don't know if you've ever had general surgery or not. I have, unfortunately, I've had several. and when you come out of surgery, you will recall that your mouth is very dry. Well, it's dry because they probably gave you atropine, which, again, dried out your GI. They don't want you choking on fluids and such. And so when you come out under general anesthetic, you're typically, again, because of the atropine they probably gave you, a little bit of a dry mouth. Of course, it works itself out in some minutes. Atropine is also used on eyes to dilate pupils. Okay, think about what constricts pupils. You have your parasympathetic system constricting pupils, growing smaller. You remove that sphincter, smooth muscle associated with your iris. And since it's parasympathetic, then the biological active molecule is acetylcholine. Well, if you drop some atropine in your eyes, it will block the parasympathetic function because it will antagonize, it will bind to the receptor and won't allow the acetylcholine to bind to it. So it dials back the effect. You may, if you go to an optometrist, and sometimes the optometrist will want to take a look at your eyes, so she may decide to put some eye drops in your eyes to widen your pupil so that she can look in your retina. And we probably have all done that. It's been done to me. And it's fine until you go outside and they have to give you the little dark glasses, waiting for the atropine to wear off so that your pupils will then grow smaller. So, again, that's something that is interesting. Let's now talk about norepinephrine and epinephrine. Okay, this is going to be dealing with your sympathetic system. So this is more parasympathetic, of course. And this is more sympathetic. So norepinephrine, epinephrine, again, is going to be your neurotransmitter. You have an array of receptors, and these are all called adrenergic receptors. So adrenergic having to do with norepinephrine, epinephrine, as you'll recall that another name for epinephrine is adrenaline. So adrenergic, that's the name connection there. So adrenergic receptors. And again, there are more subtypes than I'm going to talk about here, but these are the biggies. So you can put them in generally two buckets. You have alpha adrenergic receptors, and then you have beta adrenergic receptors. And then within the alpha and within the beta, you have subtypes, alpha 1, alpha 2, beta 1, beta 2, beta 3, etc. There are more. These are the biggies. So let's walk through and see on what tissue and what organ systems you're going to find these various subtypes, because again, norepinephrine is not norepinephrine, but what it does depends on the type of receptor it binds. The adrenergic receptors are all metabotropic. So they're going to cause a cascade of cellular events when they bind. So first up, we'll just for giggles change colors, alpha 1 adrenergic receptors. And they're going to have approximately the same equal affinity for norepinephrine and epinephrine. Again, approximately. Now, let me pause here and talk about what it means for a ligand to have an affinity for a receptor. Let me tell you what doesn't happen. What doesn't happen is the ligand will bind to the receptor, it locks in and never lets go. That just doesn't happen. That's not how you control a system. You only have one way to go with that. So ideally what you want is you want a biologically active molecule, in our case neurotransmitters, to bind to a receptor, do its effect, and then disconnect. Because as long as it's bound to the receptor, it's going to have its biological effect. You want it to disengage too much of a good thing, you understand. So all of these neurotransmitters will bind to a receptor and then will let go of the receptor. Bind to the receptor, let go of the receptor. Okay. Some receptor ligand combinations or receptor neurotransmitter combinations, the affinity is such that when a neurotransmitter binds to a receptor, it is held in place for a longer period of time. So if you had a weak affinity for a neurotransmitter and receptor, it would bind to the receptor and then quickly release. If it is a very strong affinity, it will bind to the receptor, stay there for a longer period of time, and then release. But as long as the neurotransmitter is bound to the receptor, it will have its biological effect. But as soon as it releases, it won't. The reason I'm setting all this up for you is so that you have a realistic understanding of when we speak of affinities. So my first step in you understanding this is understanding affinities has to do with time on receptor. Greater affinity, greater time. Less affinity, less time. Because there is another factor that comes into this, and we're going to see this in multiple contexts. The effect of the neurotransmitter or any biological molecule, drugs, anything, they have an affinity for a receptor. The other factor is how much of it you have. Because if you have a lot of something, then that means the probability of that molecule binding to the receptor is greater than if you have less of it. So I could have something, and I might go silly numbers here, something that is relatively strong affinity, say molecule A, and then I have molecule B, and it is a weaker affinity. And suppose they have different effects on the receptor. So if molecule A and molecule B are of equal combination, excuse me, I misspoke that, or of equal concentration, if you have as many of A as you do B, and they're going to bind to the same receptor. But if molecule A has a very strong affinity and molecule B has a weak affinity, then if you look at the dynamics, molecule A will dominate because most of the receptors will be docked with molecule A. You'll still have some B docking, but B is weaker, so it will be less time on the receptor. So therefore, the dominant effect will come from A, although you'll have some B influences. Occasionally, a B will slip in there and spend a little time on the receptor as well. Okay? So again, rolling back our conditions here, if you have molecule A, strong affinity, molecule B, weak affinity, and they have equal concentration, then the dominant biological effect will be from A. Okay, so let's run another scenario. Molecule A, molecule B. Molecule A, strong affinity, molecule B, weak affinity. But suppose I have 1,000 times more concentration of molecule B than I do A. Oh, that changes things. Because I have so much of molecule B, the probability that a molecule B will be bound to the receptor is much greater than the probability of molecule A. Even though molecule A spends more time on the receptor, you got so much of B, it will bind, release, bind, release, bind, release. But if you have 1,000 times more concentration of B, then B will be the dominant biological effect. So you always have these two things at play. What's the affinity, and then what is the concentration of these molecules before you can have a sense of what the downstream effect will be. But you always have both of those things going on, you see. So we'll run into this dynamic several times throughout the course. Okay. All that to go back here. So alpha 1 adrenergic receptors, equal affinity for norepinephrine, epinephrine, usually stimulates the tissue. Talking about smooth muscle contraction, that's typical, in the walls of blood vessels. So now I have these receptors, alpha adrenergic, embedded in smooth muscles associated with blood vessels, and it will cause them to contract. This also includes coronary arteries. So the binding of alpha 1 adrenergic receptors will cause the coronary arteries to contract. You'll find them in the urethral sphincter and will cause contraction. Erector pili muscles, your goosebumps, will cause contraction. This receptor can also bind dopamine. I'm going to come back to that later. So I have another neurotransmitter that can bind alpha 1 adrenergic receptors, and that molecule is dopamine. I will return to that later. All that to say then, you can have a receptor, but you can have multiple types of molecules to bind to that receptor. So that's alpha 1. Alpha 2, let's just change colors. Alpha 2, adrenergic receptors, again metabotropic. Again, approximately equal affinity for norepinephrine and epinephrine. These are involved in the constriction of the sphincter of the GI tract. Okay. Involved in the inhibition of insulin release in the pancreas, and involved in the induction of glucagon release from the pancreas. So this is where we're going to find our alpha 2s. Again, very much involved in smooth muscles of the GI tract and then pancreas and such. Again, there's more things involved in alpha 2. I'm just talking about some real basic, simple things. Okay. So these are my two basic alpha 1, alpha 2, two basic alpha adrenergic receptors. And again, I'm going to polish on this a little bit later when we go into different organ systems. Okay, so let's take a look at the betas. So we got beta adrenergic receptor, again metabotropic, two common subtypes, beta 1, beta 2. Beta 1, equal affinity, norepinephrine, epinephrine. Found mostly in the heart. Ooh, there you go. Now note, we had alpha 1s, they were found on the blood vessels of the heart. Here, they're on heart proper. And when bound, they will cause the heart to beat more forcefully and more rapidly. This receptor also binds dopamine. And then finally, we'll go with beta 2. Now, alpha 1, alpha 2, beta 1, roughly equal affinities for norepinephrine, epinephrine. Now, if you dig into the literature, some of the literature will say, well, one's a little more than the other. The roughly equal affinity is a ballpark as far as physiological significance. You're fine just to say they're roughly equal. Although you'll dig into it and find literature that makes it just slightly other. The bigger one, though, that is physiologically significant is the beta 2. The beta 2 has a much greater affinity for epinephrine compared to norepinephrine. Okay, so that one is physiologically significant. Beta 2 is mostly inhibitory. Okay. Found on skeletal muscle blood vessels. Found on coronary arteries. Oh, okay, so we have beta 2s on coronary arteries, and they're going to relax because they're inhibitory. Remember, we had alpha 1s on coronary arteries, and they're wanting to constrict those coronary arteries, so now we have a little tug of war going on here, possibly. So these beta 2s typically cause vasodilation, therefore increase blood flow to muscles and heart during sympathetic stimulation. These beta 2s can also be found in smooth muscles of the bronchioles. That's important because, again, I'm wanting to relax the smooth muscles of the bronchioles. I'm in a sympathetic moment, bear in the woods. I'm bathing my lungs with epinephrine, norepinephrine. Again, the adrenal gland is releasing all this epinephrine in from the medulla, again, causing my bronchioles to relax because I want to move more air. So because of this, there is a clinical point here, and the clinical point is if a patient has asthma. Now, we'll talk about asthma later, but asthma is just this generalized term. There are lots of causes for asthma. A lot of it's idiopathic. We don't know. But it all kind of comes to maybe spasms of the smooth muscle and the bronchioles that causes it make it harder to breathe. And so you want to do something that's going to cause those muscles to relax. So wouldn't it be lovely if you could find a molecule that would bind to the beta 2s and that molecule be an agonist? And so a common inhaler is albuterol. It is a beta 2 agonist. And it helps dilate the bronchioles, an inhaler, a nebulizer, and you squirt and you breathe in, and it does a lovely job of tapping down an acute episode of asthma. It's not very useful long term. Longer term, you're going to want to be put on a steroid. It's an anti-inflammatory to help with asthma type things. But if you have an acute event, then something that you inhale that is something that's going to interact with the beta 2 is quite useful. Another drug, propranolol, that is termed a beta blocker. A beta blocker will block both beta 1 and beta 2 adrenergic receptors. Let's go back, beta 1, beta 2s. Here we are. These are the two in terms of what they do. Okay? So what do these betas do? Well, they're working on increasing blood flow to the heart. They're beta 1s, going to stimulate the heart to beat more forcefully, etc. So if I block beta 1, beta 2, I'm basically interfering with the sympathetic interaction of lung and heart. And the focus on here, though, is mostly heart. So a beta 2 agonist, propranolol, blocks the sympathetic stimulation of the heart. So why would you ever want to do that? Well, if patients have coronary artery disease, which means they're having compromised blood flow to the muscles of the heart, anytime they exert themselves and they ask the heart to pump more blood, work harder, if the metabolism of the heart is such that it is working beyond its own blood supply, because every time the heart beats, it feeds itself blood through the coronary arteries. And you got a left coronary artery and a right coronary artery popping off the base of the aorta. So when the heart beats, it literally is feeding itself blood. But if you have occlusions of those arteries, so now you've compromised the blood flow, then does that not interject a metabolic ceiling where you can't work beyond your own oxygen supply as you feed yourself blood, right? So people that have coronary artery disease, if they exert themselves too much, their heart works beyond its own blood supply, and the response to that is a lot of pain called angina pectoris. And also it can be pronounced angina. You see here it both ways, angina or angina. Literally means pain of the chest. Or you could have some referred pain. It could be pain to one of the extremities. And it's very annoying. So what you do with that patient is you can put them on a drug like propranolol, a beta blocker, And the beta blocker will stop the stimulation of the heart, therefore keeping the heart from pumping beyond its own ability for its own blood supply. Well, of course, if you think about that, well, then what are you supposed to do if you start running around the block? And the answer is you don't. This only works if you have a lifestyle issue where you take it really easy. You're not asking your heart to do anything it can't do anyway. So someone who has this situation, they're on a beta blocker, they have to limit their activity, but it does help manage the pain. Okay. So, again, we're going to be visiting this a little bit more when we do cardiology. I want to get to dopamine because I'd mentioned that. Dopamine, a very common neurotransmitter. Like the receptors of these others, you have multiple dopamine receptors. One common one's a D1. You got D2, etc., but I just want to focus on the D1. The D1 receptor, you'll find these in your autonomic system. It can be found on your renal or your kidneys. It can be found on GI. Could be found in coronary vessels. And typically it can dilate those vessels. Okay? Now this gets back to the dosing issue that I spoke of earlier. Administered at low doses, the dopamine causes increased renal perfusion. You're vasodilating blood vessels, increases blood flow to the kidneys, and will work as a diuretic. Now, a diuretic, as you may know, diuresis means causing you to lose water. So if you take something that is a diuretic, it's going to cause you to increase urine production. You'll end up losing fluid. So if I increase blood flow to the kidney, that tends to cause the kidney to eliminate more fluid out of your body. It also can cause sodium loss in addition to water loss. Okay? That's low doses of dopamine. Dopamine can bind in addition to D1. Now these D1s, these are going to be the vasodilators. Again, when the dopamine binds to D1. But this is low dose. Dopamine can also bind to alpha-1 and beta-1 adrenergic receptors. But the affinity for alpha-1, alpha-2, alpha-1, and beta-1 receptors is not particularly great. So at low dose, the dopamine really doesn't have much of an effect on the alpha-1 and beta-1s. It's mostly the effect is on D1 receptors. But as you raise the dopamine level, so an intermediate dosage, and I'm not going to give you numbers in terms of low, intermediate, and high. That's not my point. It's a useless number to remember. I'm just wanting to get the concept here. If I raise the dosage of the dopamine now, I'm going to have it interact with beta-1 adrenergic receptors. And this will cause increased force of contraction of the heart, increased heart rate, and raises blood pressure. So dopamine at a higher level begins to interact with beta-1s and mimics the effect of a norepinephrine on beta-1 as well. So again, it's going to cause these results, increased heart rate, raise the blood pressure, etc. If you really go high with the dopamine, high dopamine dosage, now it begins to interact more with alpha-1, which tells you what? That the dopamine has a higher affinity for beta-1 than it does alpha-1. So I need more of it to start having an effect on alpha-1. So go back to my earlier explanation in terms of affinity versus concentration. So at high doses, not only will it be working on beta-1, it also will start working on alpha-2. And this will be body-wide vasoconstriction, including kidneys, which can reduce urinary output. So at low levels, dopamine increases urinary output. At very high doses, it actually has the opposite effect. Again, has to do with dosing, you see. So at high doses, dopamine is a very powerful vasopressor. So I'll lay this word on you. A vasopressor. A pressor is anything that is going to be increasing heart rate, blood pressure. So dopamine at high doses can be a vasopressor. Epinephrine is a vasopressor. Norepinephrine is a vasopressor. So in clinic, when someone is undergoing, say, circulatory shock, where you have a drop in blood volume, therefore a drop in blood pressure, and you're desperately trying to reestablish perfusion pressure to the major organs, one of your tools in your tool bag is hitting them with a vasopressor. And again, protocols are worked out, but sometimes you would go for dopamine, sometimes you would go for epinephrine or norepinephrine, again, depending on the situation. But these are drugs that you would use that would cause a very powerful vascular constriction, increase in heart rate, but if you're losing blood or you have some type of shock issue in terms of your vasculature and you want to reestablish blood pressure, a quick way of doing that is hitting them with one of these suppressors or a combination. So dopamine is extremely powerful vasoconstrictor in an increase in heart rate and cardiac output, and comes in quite handy in emergency situations. Okay, so we will end this block right here. This is a lovely summative diagram regarding your sympathetics and your parasympathetics. I'll let you work on this for a little bit in your study, but let me just point out some things. Over here, we have the sympathetic system. Over here, we have the parasympathetics. The sympathetics, again, I hope you have looked at the chain ganglia, but here's the chain ganglia right there. That's that little guy right there. It starts right at the base of your skull in the soft tissue of your neck and goes all the way down into your pelvis. So each one of those ganglia starting at T1 is named ganglia, T1, T2, T3. The ganglia that is rostral to T1, you only have 3 of them. You have a superior cervical ganglia, and that's going to be way high up in your soft tissue of your neck. You have a middle cervical ganglia, and then you have an inferior cervical ganglia, and then you're going to start T1, T2, T3, all the way down to the sacral area. Now, there is one aspect of this drawing that is deceiving. It leads you to believe that you only have 1 chain ganglia, and you don't. You have 2. It's a bilateral structure. They only have 1 drawn in the diagram for schematic reasons. So again, recapping some fundamental points here, and that is your entry point of your sympathetic system into the chain ganglia all happens between T1 and L2, because this is where your intermediolateral cell column is, your IML. And again, it is also a bilateral structure. So all of these preganglionic fibers will enter the chain ganglia. Now, this is additional to what I told you in the earlier lecture when we drew that cartoon out with all the various fibers and stuff. So I'm about to give you some new information. And that is when these guys enter the chain ganglia there, they may or may not synapse at that level. Some of them will enter the chain and then will ascend, because again, I have a superior cervical ganglia here, and so my T1 neurons that are sympathetic, that are part of the IML, they're going to be talking to the superior cervical ganglia, so they're going to go in, and then they're just going to ascend up and up and up and up and up, and then they will then synapse onto the neurons in the superior cervical ganglia, you see. Or the same thing down here. You could have, again, some neurons down in L1, L2, and they'll enter the cord, but they may go down to synapse onto a neuron down below it, and then that neuron then gives rise to its nerves. So it's not the case that these guys go in and stay at that level. They could go up, they could go down, or they could stay at that level. But the entire chain ganglia has to be serviced, but it's only serviced between T1, L2. So again, many of these fibers ascend or descend in the chain ganglia. The superior cervical ganglia is going to be mostly involved with your sympathetics of your head. Your thoracic organs are going to be some from that superior cervical ganglia, and you have middle cervical, inferior cervical, and maybe some T1s. Most of those are going to be your sympathetics to your heart and your lungs. And then when you get T2 and below, those are servicing the sympathetics of your organs below your diaphragm. And again, recall the scheme here, and that is the sympathetics that are servicing your body cavity in your thoracic cage. Those are going to be post-ganglionic fibers. Post-ganglionic. Okay? So the post-ganglionic here is going to be your ... Also, the fibers, sympathetics that are servicing your head will be post-ganglionic as well, because they're all coming off that superior cervical ganglia. And then note we have a big shift when we get to organs that are below the diaphragm. Here, recall that these are still pre-ganglionic. They did not synapse in that chain gang. They just passed right through. And remember I said they go and find their own ganglia. So I have a celiac ganglia down here. I have a superior mesenteric, inferior mesenteric. Again, I'm not asking you to learn the names of those ganglia. You'll learn those in gross anatomy. But these are going to be the ganglionic targets, and then they will then send out their post-ganglionic sympathetics to meet whatever organ they're going to service. Now, what's missing in this picture is going to be the scheme going to your skin and your skeletal muscles associated with your limbs. So that's not really in the diagram, but that's okay. You can imagine it being there. If you go on to the parasympathetics, again, we have our 4 cranial nerves, 3, 7, 9, and 10 in the brainstem. And 3, 7, and 9 are going to be associated again with head. But note, these are pre-ganglionics, and they're going to find their named ganglia in the head associated with said cranial nerve, and then they will then go to their targets. And then this one here, that's the biggie, cranial nerve 10. Okay, and that little guy right there goes down and wanders throughout the visceral organs, hence its word vagus. And it will supply the GI down to about the splenic flexure, and the splenic flexure, again, is going to be roughly about there. So they're going to synapse onto their parasympathetic ganglia that's buried. Again, they're small, and they're buried in the walls of the digestive tract. And then their post-ganglionic parasympathetic fibers are rather short, because the ganglia are very near the organs themselves. And then we have, picking up down on the sacral area, 2, 3, 4. They're going to be sending out their pre-ganglionics, and they're going to find their ganglia near the organ, and they're going to take care of part of the digestive tract, and then pelvis. Okay, the bladder is just just the representative organ of the pelvis, you see. So again, this is just kind of a nice summation in terms of how the various parts of your body are serviced with these autonomics. And so again, you pay attention to pre and post ganglionic and such. But I think this is it kind of pulls a lot of things together for you, I hope. Okay. Next page here. Get a little frozen up here. There you go. There you go. This is a little wordy, and I'm not going to read all this to you because most of this is just a rehash of what I have already told you. So I'm just going to ask you to read through it. Again, it's just summative things. I don't think there's anything earth-shatteringly new here. So again, I encourage you just to read through this if you don't mind. Again, I'm not interested in you memorizing celiac, superior, inferior. Again, I'll let that be worried about in gross anatomy, but I'm just telling you this is what happens. Okay, so I do want to get to this drawing here because this drawing is helpful. Again, we have here brainstem, in this case the medulla, and then we have the spinal cord here. And so this represents, this is going to be the IML. IML is going to be, let's see what I'm looking at here. Acetylcholine. Yeah, okay. IML. This is going to be your T1 through L2. I mean, the purpose of this drawing is not to represent that. The purpose of the drawing is actually drawing its focus on the receptor. So let me kind of get to the point here, which is whether it's parasympathetic, would be vagus up here. It's going to release acetylcholine. It will bind to a nicotinic receptor. Acetylcholine in the sympathetics. Nicotinic receptor, nicotinic receptor, nicotinic receptor, nicotinic receptor. Okay, so that's important to note. Acetylcholine binds a nicotinic receptor, which means it is inotropic, which means it's going to allow sodium to rush in, causing, and again, action potential down there. Okay? So now we move down and look at the postganglionic fibers, and again, up here is going to be your parasympathetics. It will release acetylcholine, as parasympathetics do, and note it binds to an M2 receptor. Now, we didn't talk about M2. I said that there were multiple muscarinic receptors, so I'm not worried about the M2 part. It's just it's binding to a muscarinic receptor, and the muscarinic receptor is metabotropic, so it's not a channel. It's just going to cause cell biology to happen in the organ. And again, find these in heart, blood vessels, and such. And it's on some blood vessels, not all, but some blood vessels. Here, now we're looking at the sympathetics. Again, norepinephrine is the biggie. Again, both of these are metabotropic. Okay, vessels, again, these are more body-wide vessels. Body-wide, they're all over. Body-wide. This is a major player in blood vessels. Okay, this is what's cool, though. Okay, this one here. Note right there, I have a sympathetic cholinergic as in acetylcholine. So now I am having part of my sympathetic system releasing not norepinephrine, but acetylcholine. And it will bind to a muscarinic receptor. And you'll find these in sweat glands. You know you sweat. Erector pili smooth muscles. Actually, the erector pili smooth muscles have a they respond to both acetylcholine and norepinephrine, but mostly acetylcholine. So point being, though, is that acetylcholine is a player when it comes to sweat glands. Vessels, acetylcholine, muscarinic on blood vessels, they found those on other animals. I'm not sure they've ever really proved that they exist in humans. But, you know, so I'm just going to go vessels. I'm going to go dogs and cats. I know they found them in dogs and cats. Humans, Maybe not. Okay, but certainly these will for sweat glands. Blood vessels depends on the animal. Okay, so we also see here that the sympathetic can release dopamine, and it will bind to the D1 receptor. Again, we talked about those for renal blood vessels. Okay, again, they can dilate blood vessels. Again, very dose dependent. And the truth of the matter is, this typically is rather minor. It's not a big player in regulating blood flow into the kidneys, but it has some effect. And then nicotinic, this is where I have my adrenal medulla cells. Again, they have nicotinic receptors. Acetylcholine binds to them, and then that will stimulate them to release norepinephrine. And epinephrine goes to the blood, and again, binding to your alpha and your beta receptors, which again are muscarinic. So again, a nice little cartoon with the emphasis on the neurotransmitters and the receptors. Okay, so finally, and again, I'm not going to read all this to you. Again, just kind of a review of some things that we spoke of earlier. Activation of sympathetic nerves to heart. Of course, you're going to have epinephrine, binds to adrenergic. Again, beta adrenergic, heart rate. Positive chronotropy means increasing heart rate. We'll talk about this later in cardiology. Positive anatropy. Anatropy has to do with how the contractility of the heart, how forceful it can contract. A positive dromotropy. Dromotropy has to do with the velocity of the electrical pulses. So the sympathetic system can cause all of these things to happen in the heart. It beats faster, beats more powerful, and the electrical system happens more rapidly. Sympathetics, adrenergics, constricts blood vessels. Okay, we've been through all that, so I'm not going to rehash that. Except for a few organs, there's a little vagal innervation of blood vessels. We've already spoken of that. And let's see, sympathetic cholinergics activates the release of acetylcholines, muscarinic receptors, sweat. Could dilate some arteries and some tissues, but again, we're talking about maybe not in humans. So you'll be careful with that one, of course. Sympathetic dopamine, kidneys, we've already talked about that. Theoretically, we see what it does. I don't know how, again, physiologically how important it is, but they're there. You certainly do have plenty of sympathetic input to the kidneys as well that causes marked alteration in blood flow. And again, we'll get onto that when we do kidney. And let's see, adrenal glands we've already talked about. Again, I've already talked about that. Basal resting conditions, we have tones. They're tonically active. And again, it's a matter of dialing one tone up, dialing one tone down to determine what the biological effect is going to be. Okay, so again, a lot of this came from CV Physiology. I love that webpage. All that's really a good read. Anyway, so this concludes our little whirlwind tour of the autonomic system. See you next time.